So, small turnout today. Everybody must be pretty well. Welcome to allergy season, right? I went working outside and had a great time working outside, but I definitely feel the pollen in the air, which is everywhere right now. If you look at your windshield, it's pretty remarkable. So, but nothing new to anybody who has allergies. Let's see. What can I say? The exam I haven't yet written. So I've got to work on that, make it as hard as I can. And what else? The material I finished covering on Friday will be the last of the material for the exam. What's that? I can't take any more. You can't? Oh, you can't take any more? Okay. Well, that's good. And I finally got that posted this morning, so hopefully you've had a chance to look at that if you need to or want to. That's that. So, what can I answer for you? Hi. Okay. Yep. 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 No. So the more what you said, her question had to do with if oh bless this thing. If I look at saturated versus unsaturated fatty acids and the effect on melting temperature, the question is what happens if you increase the number of double bonds. The more double bonds you have, the lower the melting point is. And the reason it is because of those bends that get put into the fatty acid as a result of the adding the double bonds. And those bends create more irregularity in the shape. And so as I described in class, that you really have to take more energy out of that to make it regular. And that's why it takes that you actually have to go to a lower temperature in order to make that become a solid. So the more unsaturated it is, the lower the melting temperature. The shorter it is, the melting temperature, the lower the melting temperature. So if you remember those two, then the other two are the opposite. More saturation and longer means higher melting points. And that's true of the fatty acids as well as the fats that contain them. Yes, ma'am. Okay, what's the difference between a glycerol phospholipid and a phosphatidic acid? Well, first of all, let me see. Pull up a glycerol phosphatidic acid for you. Phosphatidic acid is a glycerol phospholipid. It is, right? So the name will tell you that. Okay, where's my stuff here? Okay, if I look at glycerol phospholipids, there's phosphatidic acid. Okay, so phosphatidic acid, you see, has a glycerol backbone. Okay, it has two fatty acids on it. That's the R1 and the R2 on the top. And it has a phosphate. Well, so if we look at the name glycerophospholipid, it's got glycerol, it's got phospho, and it's got lipid. So it is a glycerophospholipid. Sure. So the question is, can glycerophospholipids be something besides phosphatidic acid? The answer is certainly. So here's an example right here. So here's a glycerophospholipid. It's got a fatty acid position 1. It's got a fatty acid at position 2. It's got a phosphate at position 3. And it's got something attached to the phosphate that has made it a phosphatide. And that's not phosphatidic acid. And that is definitely a glycerophospholipid. Yes. I'm sorry? Well, I think the general things that I've talked about with respect to their structure, yeah. But I'm not going to memorize. You're not going to memorize. You're not going to draw structures of phosphatidic acids. Yeah. Yes. Yeah. Can I go over how or why? Not why, but maybe more over how of cholesterol widens the range of the transition temperature. If I can find where I put transition temperature, I'd be happy to do that. put that? Oh, here it is. Here it is. Okay. No, that's not it. I can't remember where I put that. I had a transition temperature figure. Well, let me try the following. I will draw one for you. There, make it go. It's rather blurry, doesn't it? Still blurry, isn't it? Playback, output, freeze, autofocus. Oh, there we go. Okay. All right. So let's imagine that we have a plot of state. Oh, yeah. Sorry. This piece of paper is stuck to the thing. So maybe somebody give me another piece of paper. That way I can move the paper around. Thank you. Okay, so if I go here and I say a plot, how's that? What's that? It's not. It's kind of washed out, isn't it? Yeah. That's a little better. Something like that, right? Okay. So I am plotting state, which is liquid versus solid, as a function of temperature. Okay? And so when I do that, I will start out with something that's a solid. Okay? And as I increase the temperature, I start to see a transition, and I become something that's liquid. Is that showing? Okay. Now, the midpoint of that transition between the solid and the liquid, okay, is what we call the melting temperature, or the TM. Okay? So it's the midpoint of the transition between the solid and liquid. Basically, if you want to think about this as being half melted, half solid, that would be fine. Water does this, for example. Okay? Now, what cholesterol does is it widens that transition. So instead of looking sharp like what these do, it's going to look more like that. Okay? So when a cholesterol is in a membrane, and then this one down here is no cholesterol. When cholesterol is in a membrane, it widens the transition region. So as you can see here, the transition region really only goes from here to here without cholesterol, but in the presence of cholesterol, it's going over a much wider range. And that's really all that it does. Make sense? Remember when we had talked about the hyperchromic effect for DNA, and I showed you that transition from double strand to single strand? It's the same kind of a thing. We're going from an unmelted to a melted state, and it's basically the same type of plot. Other questions? Yes? Yeah, I didn't get a chance to finish that up, and I kind of kicked myself for that. So I started to talk about that, and then I didn't. So I was actually going to announce it in class tomorrow, but I'll tell you here. So, and then I'll also remember to do that in class tomorrow. So, prostaglandins are important for, they're a very odd group of molecules. And as I said, what happens with prostaglandins is that they're involved in the pain response, they're involved in muscular contraction, they're involved in a variety of phenomena. But one of the things that makes them important for blood thinning considerations is that prostaglandins are precursors of a group of molecules called thromboxanes. That's T-H-R-O-M. You will need to know this for that. That's why I'm going to announce it tomorrow. Okay, so thromboxanes. All right, this is very simple, actually. Okay, thromboxanes make blood platelets sticky. So what happens is they favor clotting. Now, I told you that aspirin, for example, can prevent the formation of prostaglandins, right, by inhibiting cyclooxygenase. So aspirin actually is a blood thinner also because by preventing the production of prostaglandins, it prevents the formation of thromboxanes. And since thromboxanes are involved in clotting, bang. So that's what I meant to say about that, and I never did get around to doing that. So, yeah, that's a good question. So the question is, are prostaglandins therefore like a zymogen because they're a precursor of thromboxanes? Zymogen, the term zymogen only refers to proteins. So proteins, an inactive form of a protein is a zymogen that has to then be activated. Okay, so the answer to the question is no, they're not a zymogen. They are an inactive, well, I won't say inactive, it's just like converting one molecule into another molecule is really what it is. Okay? Yes, ma'am? Mm-hmm. No, prostaglandin is a precursor of thromboxanes. Yeah.
How does the prostaglandin into thromboxane?
They're just an enzyme that converts prostaglandins into thromboxanes. Yes, ma'am.
What are the three essential fatty acids like the linoleic acid,
Okay. And linoleic. So there are three fatty acids that I said are essentially essential, okay, for our diet. And any fatty acids that have double bonds out past position 9 are ones that we can't make. So therefore, we have to have them in our diet, which is why they're called essential, okay? So they're the ones that are common that have double bonds further out than position 9 are linoleic acid, that's L-I-N-O-L-E-I-C, linolenic acid, which is just like linoleic, except for it has an N between the E and the I, and then arachidonic acid. Mm-hmm. So two of them start with L. So that's fine. Two of them start with L, linoleic and linolenic. The only difference is an N. So the linolenic has an N between the E and the I, and the linoleic is missing that N. Okay, one more time. So linoleic, L-I-N-O-L-E-I-C, linolenic, L-I-N-O-L-E-N-I-C. Okay? Yeah. Turn that thing off. Come on. And let's see. Display. Desktop. What it's doing. Okay, all right. Anyway, I'll mess with it later. What else? Yeah. Okay, so her question, actually, it's a very good one. So she says that arachidonic acid is an essential fatty acid, comes from our diet. We make prostaglandins from arachidonic acid, so if we didn't have arachidonic acid in our diet, would we not make prostaglandins? And the answer is unfortunately more complicated than that. So I lie a little bit when I say prostaglandin is essential, because if you have the other two, then you can actually make arachidonic acid from it. Okay, so yeah, but in theory, yes, that would be the case. Okay. Yes, ma'am.
For carbohydrates, when they're positioned outside the cell, does the fatty acid that anchors them, does it need a protein? Like, does it, like, is the carbohydrate attached to the fatty acid or the protein? Like,
Okay. Okay. So I think what you're asking really relates to this one figure that I showed, Mariah, and that is. Yeah, let me answer the question with a figure if I can find the figure. Lipid bilayer. That's not it. Saturation, gel, butter, membrane proteins. Here we go. Okay, so your question has to do with the carbohydrates. What are they attached to? Is that, am I understanding that correctly? Okay. So carbohydrates are generally attached to proteins in the membrane. Okay. So those make things we call glycoproteins, okay? But that's not exclusive. We can have you can see, for example, if I point to the glycolipid up here, right? That blue thing is a carbohydrate, and in this case, it's attached to a fatty acid that's in the membrane. So it's not an absolute. I'm sorry? So carbohydrates on the surface of cells frequently are identity tags. That's correct. And so they're involved in blood typing. They're involved in tissue rejection and transplant rejection, things like that, yes. Good night. Yes? That's a very good question, actually. Supercoiling is not inherent to any particular type of DNA. But interestingly, supercoiling may actually cause DNA to flip into one of those. I didn't talk about this. It may cause DNA to flip into one of those forms. So if you put too much tension in one direction on a DNA molecule, if it starts out in B to relieve the tension, sometimes what it will do is it'll flip into Z, which is the backwards configuration, to relieve the tension. So, but supercoiling per se is not unique to any particular type of DNA. Okay? Sure. So our question was, how would I add or take away turns from, to cause supercoiling? Imagine I've got a circle of DNA, two completely interwound strands, and it goes all the way around, right? So if I were to cut one of those strands, not both, but just one strand, and I would sort of unwrap it a few times and then put it back together, I would have changed the number of turns that was in that circular DNA, right? But I have the same number of base pairs. So the base pairs per turn would have changed, and that's what gives the tension that causes supercoiling. That's how I would do it. And that's how enzymes do it also. That's another very good question. Does supercoiling only happen in circular DNA? It turns out the answer to that is no, although the circular is easiest to understand. Because with circular, we think about, well, we close it back together, there's no place to relieve the tension, right? So it just stays there. It doesn't go sprawling, and release everything. It turns out if you have a long enough stretch of DNA, like a human chromosome, okay, that the ends are so detached from the middle in terms of the tension and so forth, that you can create all kinds of tension in the middle, okay, without having the ends just sort of relaxing all that, because they're just too far away, they're too big, okay? So you can have it with linear as well, but in general, it's easiest to understand with circular. Yes, ma'am. Okay, so in base excision repair, what cuts the strands? All right. So I haven't said what cuts the strands there, okay? So there are various nucleases that are involved in all of these things, and that's getting into too much detail, really.
And then, can you go over the difference between the extranuclear excision and the basic excision repair and the mismatch? I just like just like running.
Okay. So she asked me to go through the difference between nucleotide excision repair, basic excision repair, and mismatch repair, okay? And the answer to that is that they do all have, they do all look similar. They do have different things that they work on. So that's how I distinguish them for the most part. So I'm not going to take apart the mechanisms by which they work, because I'm not expecting you to memorize the mechanisms by which they work, okay? I told you that they all have nucleases that cut out pieces, and then a DNA polymerase comes in and fills it in, and DNA ligase then seals everything together. If you know that much, you know the mechanisms. The way I distinguished them then was how the different things that they worked on. So for example, nucleotide excision repair works very well on damaged DNA, like you see in thymine dimers. Okay? Base excision repair is typically invoked when you have damage or a problem with a specific base, one piece of something in a DNA. Okay, that could be chemical damage, could be something that just shouldn't be there, etc. But it's usually very specific. The mismatch repair arises from, as I said, a mismatch. So you've got something that's a C paired to an A, for example. That's a mismatch. It typically happens because the proofreading didn't catch it, but it could also result from something like I mentioned in class, the deamination of cytosine or something like that. Okay, but in any event, if you have a mismatch that's in the DNA, then that mismatch will be repaired by the mismatch repair system. Okay? Go for it. You have that look in your eye? That's fine. I'm sure everybody's happy to. I'm happy to be here as long as you guys want.
And then can you go over? I'm having a hard time connecting what methylation is and like is it good, bad? Is it like I just
Okay. Okay. So what is methylation of a base in DNA? Okay. So I only talked about that briefly. So let me tell you what I basically said. So there's Gesundheit. There's two places where I talked about methylation, but I'm going to keep it simple here, as I will on the exam. Methylation of cytosine is an important consideration in eukaryotic cells. Okay, that allows a cell to turn off transcription for a gene that's located near that. Okay? So it's a way of controlling transcription. And the methylation of cytosine is actually potentially a problem with respect to mismatch repair. Because when you put a methyl onto cytosine, it looks like, okay, it is, well, I shouldn't say it looks like, it can readily lose its amine group and become thymine. So if that happens, you've created a mutation. And at a low frequency, that actually is happening in you right now. That you have methylcytosines that your cells have created to stop expression of certain genes. And some of those cytosines have lost their amine and they become deaminated, right, which results in the production of thymine. Mm-hmm. So that's a problem. Now, well, yeah. I don't want to think about it typically as things as good or bad, but there are consequences certainly of mutation that would happen. So that would be a detrimental thing that would happen. The ability for a cell to control a gene, on the other hand, is important. So I don't want to put it in one term or the other. Yes?
Does mismatch repair have the ability to go back and fix that specific change in the cytosine thymine, or is it only when the DNA is being replicated?
That's a very good question. So his question is, does the cell have the ability to repair that thymine that appears as a result of this process, or does this only happen during the replication process? The answer is if the cell detected it as a mismatch, which it normally would, then yes, it would go ahead and repair that. And it would use that methyl information that I talked about, that somebody asked me about. I said you weren't responsible for, but I said how the cell was able to tell which strand was the newly replicated one. Okay, it would use that information to remove the T and replace it with a C. What causes it not to see that change? Yeah. Well, all of these processes have a certain efficiency with which they work. You saw, for example, that DNA polymerase worked with a pretty high efficiency, but it wasn't perfect. So there's no repair system that's perfect. This one's pretty good. If you don't read too much into what I just said, but in terms of if it catches it, okay, basically it's going to catch it. Okay. Yes. Do any of the repairs take the backbone out, meaning the opposite strand?
Like, no. Yeah, I don't know. It just takes the base out, right?
So when we look at the repair systems, all the repair systems are only going to mess with one strand, because otherwise if they messed with both, then they would have nothing to copy, right? So if I have a thymine dimer, that's going to occur in one strand, not the other strand, right? So I want to mess with the strand that's got the thymine dimer and leave the other strand alone. Because if I mess with that other strand, then I would basically, I wouldn't know where to start. So be careful with the phrase backbone. So I want to call it the other strand, okay? Because both strands have a backbone. That's the phosphodiester bond, right? So in the damaged strand, it's messing with the backbone. It's taking that stretch out, right? Right, yeah. Yes?
Looking at the allosteric regulation,
Yes.
do we call an effector any molecule, so in the ATCase, like the CTP and the substrate acid?
Mhm.
They're both effectors?
Okay, good question. So her question has to do with what is an effector when we talk about allosteric regulation. So the answer is an effector is any molecule that binds to the enzyme and affects it. So that would be regardless of where it binds it. So ATP, CTP, and aspartic acid are all effectors of the ATCase enzyme. Yes, ma'am.
In the conservative model, where the enzyme is just going back from R to C,
Mm-hmm.
is it going to be the same enzyme that binds? Because like in the sequential model, does the enzyme that binds going to be different depending on if it turns into an R or T state?
Okay.
Does that make sense?
Sort of. Okay. So her question has to do with the concerted model, and she's concerned about the flipping of the R and T states, because in the concerted model there's nothing that causes the flipping. Right. So the question is, in the sequential model, it's very easy to say, look at this one enzyme here. This one enzyme, if I put, let's say, an ATP in it, I see this one enzyme flips into the R state, and therefore I can track what's happened with that. Right. However, if I'm looking at the concerted model, I can't focus on one enzyme because I'm talking about the flipping in general, right? So yes, I could follow one enzyme flipping to T and R, okay? So it is possible to do that, yes. But in general, when we're studying enzymes, we're studying collections of enzymes. So we would think of that collection of enzymes flipping R and T, okay? And in the concerted model is always the hardest one to understand, okay? In the concerted model, the enzymes are flipping back and forth between R and T. And the question is, what do they get locked into place as? So if they're in the T in the case of ATCase, if it binds to CTP, that's going to lock it in the T state because CTP will turn it off. Mm-hmm.
But I thought that what determines if it's in R or T state is just what it happens to be in at that time.
That's correct. That's what I just said. Okay. Well, now wait. Let's make sure that you understand. Okay. I hear some resignation there in your voice, so I want to make sure I'm being clear.
Like, the way that I understand the conserved model is it doesn't matter what substrate it binds to, it's just whatever.
That's correct.
Whatever,
It doesn't.
it's just whatever happens to be at that time.
So, in the concerted model, the enzyme is flipping all by itself between R and T. So if it binds a negative effector when it's in the T state, it's going to be locked in the T state. If it binds a positive effector when it's in the R state, it'll be locked in the R state. So if we look at ATCase, we could say when ATCase is in the R state, if it binds to ATP, it locks it in the R state. If it's ATCase and it flips to the T and then it binds to CTP, it locks it in the T state. Is that making sense? The difference, and the only difference between these models is that in the sequential model, we say that the molecule causes the enzyme to flip. In the concerted model, we say the flipping occurs independent of the binding, and all the molecule does is it locks it in that state. Okay? And remember, they're just models. They're just ways of explaining things that we see with enzymes. Mm-hmm.
The quaternary interaction can cause the enzyme to flip to R or T.
Mm-hmm.
What is like quaternary interactions mean? Like,
Okay.
like, does something bind to it or not? Like,
Okay. Okay, so a question had something to do with something I had written about quaternary interactions relating to the flipping of R to T. Okay, and vice versa. So quaternary interactions, of course, relate to interactions between multiple subunits of a protein. So in the case of ATCase, there's actually 12 subunits of ATCase, 6 regulatory units and 6 catalytic units. Okay? So if I affect one of those subunits in ATCase, I can affect the subunits around it. Those are quaternary interactions. It's probably easiest to see it where I talked about hemoglobin. If you think back to hemoglobin, we had 4 subunits, and I said the binding of oxygen by one of the subunits caused the others to flip into the R state, right? So that's a perfect example of quaternary interactions, okay? Yes? Okay, Operon? Mm-hmm. Operon, yeah.
Why is it if there's too much tryptophan, then transcription stops?
Yes.
Why doesn't transcription stop? All
Okay.
mRNA that was made get translated?
Okay, so her question has to do with the synthesis of tryptophan from the tryptophan operon. And the question was if transcription gets stopped, then why doesn't translation stop, if I'm quoting you correctly?
Why is it enough to stop translation, not
I'm not sure I understand. So the only way, we haven't talked about translation yet, so don't want to get too deep here, but suffice it to say that if there's a messenger RNA that can be translated, it will be translated. Okay, so the place where transcription and where I've actually talked about translation with respect to the tryptophan operon was actually in the attenuation. And so attenuation uses transcription and translation to signal how much tryptophan is present. That's really all that they do. So if they signal that there's abundant tryptophan, then transcription will be aborted. Okay? So in the process of starting the whole process, getting everything going, if those two together say that there's abundant tryptophan, then it will be aborted, which means the whole operon won't be made. It'll stop right there, okay, very early. If they signal that it is scarce, then the entire operon will be made, and all the products will be translated as well. Does that help? So if a messenger RNA is made, it can be translated, certainly, yes. Mm-hmm. Yes? Okay. Mm-hmm. So the question is, what is the operon? Is that what is there, or that's what's being made? By being there, the operon refers to an organization in DNA, yes. Okay, so if you think about that, that's what's there. That's what an operon is. So the definition I gave you for an operon was a collection of genes all under the control of a single promoter. So those all are located in DNA, and so the operon is not the RNA that's made, no. Okay? Yes, ma'am. Mm-hmm. Oh, that's a good question, actually. So our question was single-strand binding protein. They protect single strands. Do they prevent the single strands from coming back together, is what you're saying, right? It turns out that single-strand binding protein actually can prevent that. That's not their main function because we have a helicase that's unwinding everything so that they don't really get a chance to come back together. But they can prevent single strands from coming back together and forming base pairs, yes. Okay? So the main thing that single-strand binding protein does is it protects the single strand. So it's protecting it from being damaged.
Oh, okay. I thought it was protecting it from binding back together. Okay.
It can, but the primary function is actually protecting it from damage.
And it's protecting the base pair alone, not the base pair and the backbone? Or is it?
Well, there's no base pair when you've got single strand.
Not base pair, but individual base.
So it's covering the whole thing, yeah, yeah. Yes, ma'am.
What's the difference between phosphatase and phosphorylase?
Oh, boy, here we go, yeah. What's the difference between a phosphatase and a phosphorylase? Okay, so you won't be responsible for this, okay? But I'll tell you, first of all, I will tell you what a phosphatase is because you're responsible for that. A phosphatase is something that removes phosphate from something, okay? A phosphorylase we'll talk about later. I briefly mentioned glycogen phosphorylase. I didn't show you the reaction for it, so I'll tell you, but you're not responsible for it here. A phosphorylase uses phosphate to break a bond. So in the case of glycogen phosphorylase, what it's doing is it's putting a phosphate onto a glucose, and it breaks that glucose off of glycogen. We'll talk about that later. Oh, yeah. Well, glycogen phosphorylase, whatever I said about glycogen phosphorylase, yes, but not the catalytic mechanism of glycogen phosphorylase. Yeah. Okay. Okay. So I think you're talking about the covalent modification that I talked about with respect to glycogen phosphorylase. And let me show you that. Okay. I think that's here. Yeah. Okay. So I was just simply showing you here that glycogen phosphorylase can exist, is an enzyme that can be covalently modified. Okay? It can be covalently modified. And my point with the glycogen phosphorylase is that if I put a phosphate onto glycogen phosphorylase, I will make it more active. Mm-hmm. If I take a phosphate off of the enzyme, I make it less active. And that's really the only point. It was just an example of an enzyme that you could covalently modify and change its activity. No. Okay. So a phosphatide, okay, is something that's a modification of phosphatidic acid. So phosphatidic acid is a glycerophospholipid. Okay. So let me go back to the figure and I'll show you that. Okay. Oop. And where's our membranes? Here we go. And so Phosphatidic acid is the guy on the left. It's a glycerol phospholipid, meaning it has a glycerol backbone, has 2 fatty acids, and it has a phosphate on it. If I put anything onto that phosphate, I create a phosphatide. That's what a phosphatide is. Okay? It's like you could kill somebody with this thing, right? You just, come here. You don't know the stuff, I'm going to beat you on the head, right? That's what the gruesome thing. I guess no worse than beating somebody on the head, maybe. So, anyway, you had either of you question? Ribozymes are found in ribosomes. That's correct. It's what? Okay. So, I'm going to talk more about this when I talk about translation, because this is all important in translation. But since you've asked the question, and since it's important that you understand those terms, I'm going to repeat that, okay? So, a ribozyme is a catalytic RNA. That's an enzyme that catalyzes a reaction. Okay? That's what a ribozyme is. Okay? A ribosome is a complex of protein and RNA that performs translation, that is the synthesis of protein. No. So we'll talk about that when we talk about translation itself. tRNA is not a part of the complex. However, ribosomal RNA is a part of the complex. So ribosomal RNA is part of a ribosome. And ribosomal RNA, one of them is a ribozyme. But you don't need to know that yet. But you do need to know what a ribosome is versus a ribozyme. A ribozyme is one of the ribosomal RNAs is a ribozyme. That's correct. Yeah. Ribosomes make protein. That's right. They're complexes that make protein. And we'll talk about that. That's what we'll talk about tomorrow, actually. Yeah.
So, I know that the carboxylation of thrombin would then bind calcium and then clot. How does that have to do with fibrinogen and fibrin?
Okay. What's the relationship between the carboxylation of prothrombin and the formation of the clot? Basically, is what you're asking. Because, okay. So let me answer that question in 2 parts. Okay. So the first part is the formation of the clot. So the clot forms when fibrinogen is converted into fibrin, and fibrin is an amazing molecule. It will form natural polymers all by itself. It forms polymers by itself, and those polymers are what form the clot. So if you want to clot blood, you have to convert fibrinogen to fibrin. Okay? Then the question is, well, how do you convert fibrinogen to fibrin? Which is part of your question here. Okay? The answer is that an enzyme called thrombin catalyzes that conversion. So prothrombin is the precursor to thrombin. And since you want prothrombin to do that, you want it to be at the site of the wound, which is why you carboxylate it so it can bind calcium and stay there.
So the binding of calcium just stays
At the site of the wound.
It won't form the clot until the thrombin
Is con-
the fibrinogen.
That's right. Fibrin is the polymer that forms after fibrinogen has been activated, yes. So vitamin K is necessary for the carboxylation of prothrombin. Nothing more than I just said, no. So I gave the fibrinogen to fibrin as an example of a zymogen. In the fibrinogen form, no polymerization, no clot. But in the fibrin form, it's active, and it will actively polymerize and form a clot. Prothrombin is a zymogen of thrombin. Yeah. It doesn't. So they don't all. Yeah. So what happened, this is what, if you look at for example, the periodic table in chemistry, okay, you see that the symbol for tin is SN, right? So those were named before we had a designated system that organized nomenclature. The same thing happened with proteins, and in this case with zymogens. So not all of them. We also have what we call proinsulin, which is the precursor of insulin in our body. It was just named before they used the OGEN system. Yeah. The blood clotting scheme is an incredibly complex. I say it's beautiful simply because through all of that complexity, our body doesn't make clots any more than it does. And it only makes them when they're needed, 99.999% of the time. And it makes them incredibly rapidly. So when you look at the complexity of the system, and I'm just barely scratching the surface with what I've given you here, it is remarkable how well it actually functions.
What's the phosphatase graph?
What's the what?
The five-phasic graph?
Five-phasic graph.
Yeah, like,
The what?
no, it's like, I think it was like when it's kind of, like, it's like the artificial bond that gets broken by chemistry.
Is that when it gets yellow?
Yeah, the color change thing.
Oh. So, I know what you're talking about. Yeah. Okay. Yeah. I thought, whoa. Okay. Yeah. Think about that. So, what that is a study of the mechanism of action of chymotrypsin. Okay. So let me pull that up. Let's see if I can remember where I talked about that. There's the molecule that the reaction is occurring on. So here's chymotrypsin, here's the molecule, and you form a covalent intermediate and you release something that is yellow. So the graph that you're talking about that shows that is actually that's not it. It's on here. That's not it. This better be it. Not it. Where was that graph? Do you remember where it's at on here? I'd be happy to show you. Oh, here it is. Here it is. This is the graph you're talking about, right? Okay. Sure. I want to make sure I'm answering your question. Okay. So this is biphasic in the sense that it's showing two mechanisms that are going on. We see a fast phase, okay, in which we see rapid accumulation of product over time, and then we see a slow phase, which is over here. And if you remember the chymotrypsin mechanism, or all serine proteases, there's a fast phase which involves the cleaving of the peptide bond. Okay, the slow phase is the release of the peptide from the enzyme. Okay, because they become covalently attached during the breaking of the peptide bond. Okay, so the biphasic here is the cleaving of the peptide bond and then the release of the peptide from the enzyme. Okay.
And then, like, what is it called when you do the, like, the artificial bond to tell, like, the color change? Like, how fast? Like, what is that called?
What's it called to make the artificial bond? There's not.
Is it like a method, or is it like a
So all we're doing with the colored molecule is just studying the two steps of the reaction. So there's no name I gave to the bond.
The yellow indicates that the peptide bond.
Has been broken. That's right. Yes? Mm-hmm. Right. So the slow part is the release of the second half from the enzyme. The first half, there's no attachment to anything, so it just flies away. And that's the part that happens fast. Yeah. Yes? Okay. That's correct. So the only place where the effector molecules come in is after the enzyme has assumed its shape, they can lock it in that position that that the enzyme happens to be in. Okay? Yes? Very good, yes. So the semi-conservative model of DNA replication says that when you replicate DNA, the progeny have one parental strand and one new strand that's been made from it. Yes? Yes? Mm-hmm. Well, like anything else that I talk about, whatever I talk about in class is the level of depth that I expect that you will know things. Yeah. Yes, ma'am.
How do the pyrimidine nucleotides and the purine nucleotides affect the HCCA process? Like I know that HCCA is a purine and CCCA is a pyrimidine.
Mm-hmm.
I don't know, like,
Okay.
how those connections cause.
Okay. Sure. So our question was, how do ATP and CTP affect the metabolism of purines and pyrimidines ultimately, which I think is the question that you're asking. So I talked in class about how a couple of things. So first of all, ATP is a purine and CTP is a pyrimidine. And so we could think of each of those as being indicators of the total purine versus pyrimidine concentration in a cell. Okay? So we could say if ATP is high, then purine levels are high. If CTP is high, then pyrimidine levels are high, et cetera. If ATP is low, then purine levels are low. CTP is low, pyrimidine levels are low. Okay? The only point I make about those levels is that cells try to balance how much of any nucleotide they have, because too much or too little of a nucleotide leads to mutation. So ATCase turns out to be a very good enzyme for balancing all of those things. If a cell has too much CTP, then CTP will feedback inhibit the ATCase, and consequently, all of the synthesis of pyrimidines will stop. So there's one way of preventing the cell from making too many pyrimidines. Okay? If CTP levels are high, then the synthesis of that whole pathway will stop. On the other hand, yeah. Okay. So a question had to do with the base pairing that's happening in DNA with a pyrimidine versus a purine. The answer is yes, but that's actually in the synthesis of DNA, and we're talking here about the synthesis of the nucleotides themselves. Okay, so you're right. You wouldn't pair a C with a T because it wouldn't have the proper shape, nor would it have the proper hydrogen bonds between them. Okay, all right. Now, on the other hand, if we have a lot of ATP, okay, then that means purine levels are high, and if purine levels are high, what's the complement to purines in DNA? Well, the complement are pyrimidines, right? So if the purine levels are high, we want to be synthesizing more pyrimidines. So in that case, when ATP levels are high, what it does is it activates the ATCase so that the pathway to make pyrimidines gets a head start. So through this enzyme, there's balance. Too many purines cause the cell to make pyrimidines. Too many pyrimidines cause the cell to stop making pyrimidines. Does that make sense?
Yes.
Yep, good. Good questions. You guys are thinking about this, yeah.
A similar pathway exists in like
So the question is, if you didn't have enough purines, what would happen, right? Well, it's a very good question. I will talk near the end of the term about individual nucleotide metabolism, and it turns out that yes, there is a way of turning on purines when their levels are low, but it's a fairly complicated process, and it doesn't involve pyrimidines, so it only involves purines. So, well, ATP is a purine, so it turns out that when a cell is making purines, okay, ATP and GTP get made by the same pathway. So if you have a lot of ATP, you probably have a lot of GTP. And if you have a lot of CTP, all the pyrimidines are made by the same pathway too. Then that means you have a lot of UTP and TTP. Okay? Yes? That was the enzyme that was doing all this was ATCase. So one enzyme is basically balancing everything there. It's a very remarkable enzyme. Yes, ma'am.
So why is tyrosine phosphorylation the most important?
Oh, okay. Her question was, why is tyrosine, you have good ears, Mariah, you listen well. Why is tyrosine phosphorylation what I described as the most important but the least common, all right? And tyrosine phosphorylation is very important for helping a cell to make the decision of whether or not to divide. It's involved in a process called signaling. Now, I'll talk a little bit about signaling later in the term, okay? But with signaling, cells are talking to each other, okay? So the way that they communicate is one of the ways in which they communicate is by putting phosphates onto proteins, and one of the places they put phosphates is onto tyrosine. So commonly, those phosphorylations of tyrosine, and again, these are things I haven't talked about yet, so you're not responsible for this, but commonly the phosphorylation of tyrosine, what's that? Well, so I'm a firm believer that I should be giving you guys as much as I know, because I really want you to know stuff, and you look at the exam as well. That's what he wants us to know, and that's I really want you to know biochemistry. So that's why I tell you some of these things in some depth. But putting that phosphate onto tyrosine has very different effects than phosphorylation of either serine or threonine. So if you think about it, this decision to divide or not is a very important decision for an organism like us. Because if we screw that process up, we form cancer, right? And so we don't want to be dividing all the time. And so one of the reasons that phosphorylation of tyrosine doesn't happen as much as the other types is because it is involved in controlling a very important process. So the cell wants to be careful in terms of how much it's turning that on. Long answer to a short question. Yeah.
And then I just have another question. So after DNA is replicated,
Mm-hmm.
and you have your template strand, and what's the other one?
Okay. So when we talk about template strand and coding strand, we're talking about transcription. Okay? All right. So the template strand is the strand that's being copied by the RNA polymerase. The coding strand is the one that's not being copied, but because it's complementary to the coding strand, it has the same sequence as the messenger RNA that's made, except for it has T instead of U.
Is that relevant to when's the old one and the new one? From it being, like after DNA replicates?
No, you're confusing two things here. So the old one versus the new one relates to the replication of DNA.
Right, but
Right.
one of those is going to be transcribed, right?
So that's correct, but that's independent of the replication. So we talk about
It doesn't matter.
it doesn't have any effect on the transcription, no. Old versus new has no effect on transcription, yeah. Yes, ma'am? First part, I didn't hear what you said. Yes. Yeah, it's a good question. So I described a bunch of proteins present at the replication fork for prokaryotes. Are those proteins also present in eukaryotes? They are. They have different names in eukaryotes, and they're a little bit more complex in eukaryotes, but for our purposes, we can say that they're essentially the same things. So rather than confuse things with more names, I haven't given you names for the eukaryotic proteins. I think the one I talked about was PCNA instead of the beta clamp. That's the only one. Yeah.
Prokaryote DNA circular have two replication points.
Yes. Yes. So the question was, does a circular bacterial DNA have one origin and two replication forks that come off of that? And the answer is yes. Yes. That screen turning off all the time is annoying. I can't seem to change it. Yes? Okay. Okay. So the question was basically the differences between DNA polymerase 3 and DNA polymerase 1. And she noted, it's Amy, right? Yeah. So Amy noted that the DNA polymerase 1 is found most commonly on the lagging strand because it's removing those primers and filling in those gaps that result from that. And the polymerase 3 is located on the leading strand where it pretty much just stays on and goes and does its thing. It also synthesizes those pieces after the priming has happened on the lagging strand as well. So there's lagging strand synthesis for the polymerase 3 as well. The question was, do both polymerases put in complementary bases? And the answer is yes, because that's necessary for the DNA replication. So they both do that. So all DNA polymerases always go 5 to 3. Okay. Because of that. So both DNA polymerases always go 5 to 3 in the synthesis of DNA. Don't confuse the synthesis of DNA with proofreading, which is a 3 to 5 process, right? Proofreading is simply an exonuclease that backs up and chews things out, and the polymerase moves it forward. But the polymerase always goes 5' to 3'. Okay?
So if there's no exonuclease in the polymerase 1, how does it proofread off the front?
So the polymerase 1 and the polymerase 3 both have proofreading. Okay? They both have a 3' to 5' exonuclease. Polymerase 3 is lacking the 5' to 3' exonuclease that removes the RNA primers. That's why polymerase 3 is not on the removing the primers on the lagging strand. That's why polymerase 1 is doing that. Okay? So can both polymerase 1 and polymerase 3 both produce phosphodiester bonds? Yes, because that's what happens when you build a DNA. Yeah.
So the things that polymerase 3 lack, the 5 to 3 exonuclease that removes primers.
That's correct. Polymerase 3 is lacking the 5' to 3' exonuclease that removes primers. That's correct. That's correct. Right. So DNA polymerase 1 has the 5 to 3 exonuclease, the 3' to 5' exonuclease, and the DNA polymerization activity. Yes. Okay? Can I think of the RNA that's put on? You mean in the primer RNA as a cap? I wouldn't like to use that term because cap really refers to what we see in the messenger RNA in eukaryotes. So I would be careful not to use that. No, I wouldn't say that. We call it a primer is what we call it. Yeah. What makes sense? Oh, the primer. Okay. Yeah. So the primer tells the polymerase where to start, and then the polymerase starts at that point and moves forward. That's right. Yep, take care. Clear as mud? Okay, looks like it's winding down here. Good. Very good questions today. You folks are thinking about this. My advice to you is, I know it's stressful, okay? Do what you can to relieve stress, because I find stress is a big barrier to learning. One of the things I tell students to do is, if you're feeling stressed for things, and you do tend to stress with exams, I generally recommend getting some good exercise the afternoon of the exam, okay? Go out and take a nice jog, run, walk, whatever it is that you do that gets the old heart pumping, because what that does, and there's a biochemical reason for this, what that does is it breaks down stress hormone, cortisol. And by breaking down stress hormone, what you're doing is allowing your brain to actually work. Stress hormone isn't there to activate your brain, it's there to activate your muscles. And so that's why people have a harder time dealing with stress when they're taking exams. So if you break down the stress hormone, you help yourself. So get some exercise on the day of the exam, for what it's worth. There's a biochemist talking to you. Okay, I'll see you guys. I will be around in my office tomorrow. Unfortunately, tomorrow I'm fairly busy. I'm always happy if I can help you there, but I will have more time on Tuesday. So you're welcome either time, but I may not be as available tomorrow as I will be on Tuesday. The exam is Tuesday.